All right, so good afternoon, ladies and gentlemen. Thank you for coming for the second part of my seminar. I'm sorry for those that did not attend the first part, but that's all right, and you can live without it. So this seminar builds on a concept that I tried to substantiate during my first seminar, and it is that in many cases it is very difficult trying to assemble in a rational way genes for building a new pathway for degradation of a new compound, following this idea of a superbug. So the superbug is this notion that you can go into the environment, get genes from various origins, get promoters, get you name it, and then in a rational way you combine everything in a single bacterium, and then you have something that has a superior catalytic ability. So in my previous talk, I discussed with you a large number, or at least some evidence that would really go against that notion. And the alternative that I proposed was the assembly of communities. So in this talk, I will re-engage in discussing not so much the concept behind assembling communities for the sake of biocatalysis, but what tools are out there, which ones we have developed ourselves to let the researchers being able to put together communities of bacteria that otherwise may not assemble spontaneously. All right, so as a reminder, these are playground, the type of scenarios that we are interested to explore. And in these scenarios that are polluted with chemicals, we often find bacteria with amazing catabolic abilities, with amazing, capacities to use compounds that for us are bad pollutants, convert them into CO2 and water, or transforming them in other interesting things. And out of many years of research in many laboratories, there's a long list of bacterial isolates that have been identified with, as I said, amazing catabolic capabilities. In this talk, as it was the case in my previous talk, I will refer only to bacteria that can be grown in the laboratory. You know very well, up to 90% or more of bacteria that are out there cannot be taken to the laboratory and make a culture out of them. But in this case, we are going to talk about the other 10%. So in this other 10% bacteria that you can grow in the laboratory, you find once in a while fast growers on compounds as nasty as toluene or phenol or naphthalene or 2,4-D and so on and so on. And many of these fast growers in these compounds belong to a type of bacteria that rotate around Pseudomonas. And the reason for that I will tell you in a moment. But it is very frequent when you go to the environment and try to isolate bacteria growing in unusual chemical structures, you come up with bacteria that belong to these groups. All right, so in our laboratory for some time, we have been interested into this type of business. I have to make a disclaimer, and it is that my laboratory is a biotech laboratory, and I have no problem to say that. So that means that we are interested to know how things work, but always for the sake of having that knowledge applied to some practical issue. So in that context, we are interested in environmental bacteria because we are interested in biocatalysis. And this is a kind of a general flowchart of our laboratory. We start with bacteria that are in the wild. They look nice, but not very manageable. Then the first step is trying to domesticate them. And then finally, and this is the transition between standard biology and synthetic biology, trying to engineer them in a very, very predictable way. So I would say that we are in this limit between domestication and engineering, as you will see in a moment. And what is bacterial biocatalysis? Well, this is again a simplification. So you have a bacterium here, you have an input and you have an output, a substrate and a product. Then something happens here, and then the substrate is converted into a product. There's a variant of this situation, and it is that once in a while the reactions are delivered by the live bacteria, but they don't happen inside, they happen outside, whether completely in the external part of the bacterium or in the periplasmic space, that region that is between the two membranes in gram-negative bacteria. So this is the general scenario, and as I told before, and I explained in detail in my previous talk, this is what we don't want for a number of reasons that you may want to see in the video if it happens to be in the web page of the program, and this is what we would like to have at the end, okay? And if you are curious, we're about to publish a kind of long review with Antoine Danchen to explain in detail why this situation has very few chances of success versus this other situation. We will have a consortium in which each member of the consortium delivers one step in a complex biochemical pathway. All right, so we have various issues here in the way to assemble artificial bacterial communities. And one issue that we have spent some time thinking and trying to solve is this problem of how to stick bacteria to each other. So you may think on a consortium like free-floating bacteria, but for the most part, if you want an efficient catalyst to work as you want, you may want bacteria to be in close contact in such a way that if one product is released from one bacterium, it finds the next partner immediately rather than just floating around and so on. So one interesting question was, can we develop some type of adhesins in such a way that I want to stick A to B to C, then I can assemble in a prefixed order and knowing in advance how it's going to be this interaction. So in the best scenario, this is what we would like to have. So we'll have to have bacteria A and bacteria B, and they have to interact through a specific adhesin that is produced or displayed by one of the bacteria on the surface, and then the other presents a target that is specifically recognized by adhesin in such a way that we have a specific contact A into B. Just by looking at this, you immediately recognize some problems that we're going to face in a minute, and is that to get that happening, the adhesins and the target have to be let interact without any impediment of structures that might be hanging or stuck to the outer membrane of one bacteria or the other. I will return to that in a moment. The other issue that one has to keep in mind is that the adhesins have to go somewhere on the cell surface, and they can go either to the poles or they can go to the general, say, intermediate surface of the bacterium. And as you may recall, in a bacterium, not both poles are equivalent. There's an old pole and there's a new pole. And once the bacteria divide, every new pole becomes an old pole. And only the new poles are the ones that appear to be in the septation part. And as you will see later, also this makes a big difference because depending on whether the adhesin is in the pole or is in the middle, you get different three-dimensional configurations of a possible consortium. All right, so then the other actor of this story I will discuss with you today is the testbed of these concepts, okay? And the testbed is this bacterium called Pseudomonas butida. We use a strain called KT2440. You may remember that Ati mentioned some features of this strain in his talk, and we are very interested in this bacterium because it comes from the environments that exactly are adapted to or are forced to run these harsh reactions that I was telling you about. So this bacterium comes originally from soil, and the natural habitat of this bacterium is either the root surface of plants or soil polluted with various chemicals. And when the genome was sequenced, then a number of interesting good traits were found that were known in advance in some cases, but also a number of bad traits were found. So the good traits, and this is what makes this bacterium very interesting for this type of engineering, is that it is solvent tolerant, it's able to live and in the presence of relatively high concentrations of organic compounds. It has a very high NADH and NADPH level, and this is very good for metabolic engineering, mostly if you are interested in reactions involving some redox steps. It has a very diverse metabolism and also is a GRAS bacterium. GRAS means generally regarded as safe. So that means that you can use them in your laboratory or in the industry with no problem. It's even less problematic than E. coli, okay? But it has also some bad traits. It has a high antibiotic resistance because of various genes and pumps that happen to be around the genome. It has a complex surface, it has many prophages, it doesn't have a real glycolysis, and that complicates metabolic engineering a little bit. It's an obligate aerobe, so that means that it requires oxygen to grow and to live, and also it has a kind of bad reputation because there are other Pseudomonas that are pathogens, Pseudomonas aeruginosa, and sometimes the industry don't want to be associated to the use of Pseudomonas, but so be it. The other thing, and this is what for us is more tricky, is that the surface of this bacteria is not smooth at all. This is a cartoon or a computer representation, but this is the type of situation that we find or you have in bacteria that you recover from the environment. So Pseudomonas have a flagella, and also it has pili and fimbriae. And you may see immediately that if you want to stick this surface to something else, then all these pili will impede interactions with other bacteria of the type. The other thing that we don't like, or is something to avoid for this type of engineering, is that Pseudomonas putida has its own attachment agenda. So it forms biofilms, it will form clusters, it has a complex way of interacting with surfaces that is mediated by a molecule called the cyclic GMP. So it's quite complicated. So these bacteria are used to live on the surface of plants, and therefore it makes sense that it has developed its own kind of mechanisms to deal with surfaces and so. So what do we do? I mean, do we have to give up, or is this an unsurmountable obstacle, or are there something that we can do about it? Okay, so what we figured out time ago is that, well, one way to go perhaps is to take a look to the bacterium and try to see whether we can avoid all these obstacles to interaction that natural bacteria have naturally. So we have flagella, we have this pili and so on. So we thought, well, maybe we can, you may call it emasculate, you can cut the flagellum here, then you can shave the kind of pili that you see on the surface, and finally you can like iron out the surface, and then by doing that, you can think that you will produce a kind of a smooth surface where you can really stick your adhesins and make the concept working. So what we have made over the years, and this is a one slide that we keep on putting genes here that we delete, but it's in fact a pain in the neck to do that. We have deleted up to 35 genes now in the whole genome of P. putida in a recursive fashion by using some techniques that we developed in my laboratory. And in one particular set of deletions, what we have made has been to focus on these surface structures that we could think that could interfere with attachment or interactions of these bacteria with other bacteria. So we have deleted adhesion proteins, LPS, some fimbriae, curly, cellulose production, you name it. And at the end, we have produced what we call the surface naked P. putida, okay? So unlike this other hairy and flagellated counterpart, this bacterium, when you look at it in the microscope, it has a apparently smooth surface. And also, we observe when we characterize the physiological characteristics of this bacterium that it has acquired some nice features. For instance, it has become more sensitive to antibiotics, and this is very nice because then you have more genetic tools that you can use for its engineering. Okay, so this is what we had at the beginning, and after all these genetic tricks, we come to these naked strain cells. Now, this is part of the problem. The other problem is when we talk about adhesins, what are we talking about? So there are various outer membrane proteins in gram-negative bacteria that you can entertain as candidates for this type of operations. But the one that we started to work with some time ago was this type of proteins called autotransporters. So autotransporters are a type of secreted proteins that follow a mechanism of secretion that is quite tricky and simplified in this cartoon, through which the whole unfolded polypeptide goes into the periplasm, and then it falls in a funny way. But what happens is that it forms a barrel, and it is able to pass a domain or another polypeptide through the barrel and display that on the surface of the cells. And in this particular case, this is autotransporter that puts on the surface a protease, and it comes from Neisseria, that is a pathogen. But in fact, you can fix genetically this situation and use this barrel only as a kind of scaffold for displaying a heterologous peptide, a heterologous protein on the surface, okay? So as I will tell you later, there are more choices for putting these adhesins on the surface, but this is the one that we started with some time ago. So this is the architecture of one of these autotransporters. So we have this beta barrel domain that gets anchored on the outer membrane, and then you can fuse here what is called a passenger domain of your choice. Obviously, nature, there are passenger domains that nature have provided with, but artificially with genetic engineering, you can replace these passenger domains by another protein of your choice, and then that protein, in most cases, not in all cases, but in most cases, we present it on the surface. So when we started to play with these ideas, and This was like 10 years ago, so it has rained a lot since then. So we said, well, let's start by something very simple. So one possible minimal adhesion, for instance, is to take a leucine zipper. So leucine zipper is a motif that appears in some proteins, in bacteria and eukaryotes and other places, and they have the property of forming dimers very, very easily. So what we speculated was that, well, if we put one of these motifs on stuck to a transporter domain, and then this is delivered to the surface, then what will happen is that after a while, the cells will be coated with this dimerization domain, and the bacterium A can be put together with bacterium B. This type of construct is interesting because these leucine zippers are very short, and that means that if we get them to work, so that means that the cells get enough proximity to really have these leucine zippers close to each other as being able to interact, what will be a kind of validation of this shaping operation of the surface that I mentioned before. So this is what happens. So well, this is the wild-type bacterium with all the hairs and the flagellum and everything. So you see that, well, regardless of whether one expresses these proteins on the surface or not, they are basically hanging around and they don't really precipitate. But when you take this naked strain and then you express on the surface this dimerization domain, they precipitate instantly, okay? So that means that, in fact, the cells have the capacity to cluster because of the display on the surface of this dimerization domain. This is a kind of blow-up of one of the experiments, very nice. So when you have a strain, well, you have cells without these leucine zippers displayed on the surface, the cells happily hang around. If you add IPTG, it's an inducer that will make the bacteria to be coated by these dimerization domains, then they immediately precipitate. And this is something that we are after. But then we want to see what is the actual structure of the potential, say, partnership between the cells that we had by doing that. So what we made was to, well, I have to make a parenthesis. A trick that you can make in this type of operations is to use different types of leucine zippers, so that in some cases they will dimerize with itself, in other cases it will dimerize with another leucine zipper of a slightly different structure. So in this case, what we made was to take one Pseudomonas putida, one naked Pseudomonas putida, labeled with GFP, green color. We took another Pseudomonas putida, tagged with mTERI, red color, and then what we did was to put together in each of them this leucine zipper construct to be displayed on the surface in such a way that the leucine zipper of one bacterium will match the other and not with itself. So when you take a look to this in the microscope and then you reveal the position of the bacteria with green or with red, you get something like this. Then this is GFP only, this is mTERI only, and this is what you get when you get these two together in conditions that make the cells to express and to display these leucine zippers on the surface, okay? And you see these clusters that otherwise would not be formed if you didn't have these leucine zippers. That's another more beautiful picture here. So for instance, a green, red, and you see this kind of rosaries. One of the things that we have not really looked into this in detail, but it looks like if the adhesins tend to go to the poles. So that's something that we have to figure out because it may have some consequences in the future to see how we can assemble three-dimensional structures, okay? But as I said before, whether the adhesins go to the pole of the rod or the bacterial rod or go to the center or go to the entire surface, maybe the final structures that we get in this artificial consortia will be a bit different.
Excuse me, what does this picture actually show? Does it show what you can recover from the sediment, or is it something?
No, this is in solution.
So this is in solution?
Yes. Right. All right. Okay, now, and this is what we would like to have at the end. So, so this is what we have at the moment, and this is what we would like to end. So by combining various adhesins, various types of adhesins in various parts of the cells, then we may be able to make this type of constructs with some catalytic abilities. But this raises a question. What I told you before has to do only with leucine zippers, and leucine zippers is nice as a model adhesive, but in reality it's just one. So we may need more to make sure that this one matches with this one and this one with this one and so on and so on. So how do we get more adhesives and partners for these adhesives? So one way to go, there may be others that we started also time ago, was to play with antibodies. So you may think that antibodies may turn the ultimate adhesives, because imagine a situation where you have a partner in an interaction, partner A, that is coated, we'll see how we do it, with an antibody that is produced or is, say, displayed on the surface, and you have another bacterium B, that you have an antigen that is recognized by this antibody. That would make the two bacteria to get together. So, but antibodies, as they appear in nature, are very big. As you know, they have the heavy chain, the long chain, and so on. And, well, obviously, you cannot dream of producing and displaying these huge structures on the surface of bacteria. However, for the last 20 years and so, there are various techniques that allow to reconstruct the business part of an antibody, namely the part of the antibody that will interact with an antigen, in the form of a single polypeptide. So this is what is called single-chain antibodies. And what it is that you can go to the sequence of an antibody and try to reconstruct in a single polypeptide the two chains or the two parts of the chains that are involved in antigen recognition. And there are a large number of tricks that have been developed over the years to have this type of structures. So years ago, and that means quite a few years ago, we said, well, maybe we can make bacteria to display these recombinant antibodies on the surface. So what we did was to make fusions between these autotransporters that we had used before for the leucine zippers and one of these single-chain antibodies. In the single-chain antibody, remember, you just capture in a single polypeptide the entire business part of a whole antibody. And this is all we need for this type of application. So this is what this is the genetic constructs, and this is what you should get at the end, an autotransporter that is producing this antibody on the surface. Well, we did that, and then when you run a gel, what you and you monitor what is the status of the protein by using an antibody that will recognize an antigen in the structure of this protein fusion, then you observe something like this. So the good news is that the fusion is produced and the antibody is in place and everything, and this is represented by this band. But the bad news is that you have all these other bands that indicate that cells are not very happy expressing this protein, okay? So this is a way to go that we could not really consider as adequate, because cells do the job, but the physiological cost of expressing this type of proteins on the surface is very high. So we went back to antibodies, and then we discovered in the literature that there are other antibodies that could be more attractive to this end. And it happens, and it came to us as a very curious and fascinating story, that camels, the animals, produce a type of antibody that are naturally single chain, okay? So whereas in the case of standard immunoglobulins, you need the concerted structure formed by the heavy chain and the light chains to produce what immunologists call the CDR, the complementary regions of an antigen, and they are formed in the tip of that Y. In the case of an camel antibody, you also have the Y, but the tip of the Y involves only one polypeptide. So that means that if you manage to get this polypeptide in place, then you have a much smaller protein, and then the possibilities of expressing them in bacteria and eventually to put them on the surface of bacteria are much higher. So this is very nice, but you need camels to take this thing, and camels are difficult to get. But fortunately, we have some islands in the Atlantic Ocean. Maybe some of you have been there for tourism, the Canary Islands, and they have many camels that they use for tourists for the most part. Formerly was used for agriculture, but now they are used for tourists. They are well fed, they look nice, and so. And we have a collaboration with a company there that has a big herd of camels to get our antibodies punched into the camels and then get them back to Madrid. And then we have put in place a flow chart to get camel antibodies from camels out of any antigen that we send to them. Well, what we do is that we inject proteins to camels. It can be one protein, it can be an entire cocktail of proteins. Then we get the blood, we get lymphocytes, we extract RNA, and with some primers that we have, then we go through a phase display system. This is something that I will not get into the details, but at the end we can really get these single-chain camel antibodies that are so good for protein engineering because they are small, but still they capture all the antigen binding capacities of the standard antibody. And once we have that, then we go ahead and clone them in the vectors that we have developed to display these camel antibodies on the surface. So this is the situation that we get at the end. We have the outer memory here. We have an autotransporter. Then we have an E-tag for monitoring purposes. This is an immunogenic peptide. And then we have this protein here, and then this antigen binding domain here. So we do the same that we did before to see whether bacteria are happy or are not happy expressing this type of fusions. Then we observe a situation that is very different of what we observe with the standard single-chain antibodies, namely that, you get a very discrete band that is produced with no indication of degradation. That indicates that bacteria are fine producing these antibodies on the surface, and that's really, really good. Furthermore, these are some controls that we made. This is a standard single-chain antibody with the two chains that are joined through a linker, and you see that cells are very happy. And this is one of these antibodies that has been engineered as devoid of disulfide bridges, and you see that cells are relatively happy as well. So this is connected with the mechanism of secretion that we will not discuss, but the final take-home lesson is that bacteria appear to be quite happy expressing these single-chain antibodies originated in camels on their surface. Well, these are some other extra experiments in which we could really see, and this is something that comes very, very as an interesting piece of information for this community structure, that once you have these single-chain antibodies in hand, you can make genetically a kind of rosary of one antibody against after the other, and to have these antibodies with different specificities, and still this kind of rosary of single-chain antibodies will be presented on the surface, okay? So this is the situation that you have with one antibody. This will be the situation that you have with two antibodies that will be secreted and displayed through the same procedure, or you can get up to 3 antibodies. We have not kept on putting more antibodies. Maybe we can put more, but at the moment we have 3 antibodies, and we know that 3 of them are secreted at display and are active against their cognate antigens. So this is a kind of a direction in our research that is very, very active at the moment, and we are exploiting it in many different directions. And this is perhaps one of the tools that we have in the future for assembling these communities, because if you have a repertoire of antibodies and you have a repertoire of antigens in different bacteria, then you can really make combinations and make models that perhaps will tell you ways of assembling three-dimensional communities. I don't know, this is something that we have to explore in the future. Now, let me give you another angle to this concept of assembling artificial communities or communities with a certain structure. And this idea that it's not enough that you put the bacteria together, they have to get some degree of communication with each other, okay? So what we have been also trying to explore in recent years is whether we can really think on not only putting together physically these bacteria, but trying to develop some type of language that they can understand to do things that they would normally will not do. So first of all, you need some kind of conceptual frame, and the conceptual frame that we started to explore some time ago is this idea of using regulatory nodes in transcriptional regulation networks as logic gates. Well, you know logic gates, the basis of computing, you can implement them through many different, material devices. But it happens that promoters in bacteria do implement logic operations, and you can translate the logic function of every transcriptional regulation unit into some type of logic gate, okay? So you have amplifiers, you have NOT gates, you have AND gates. I mean, you can really make a translation of regulatory events that biologists would describe in a soft language into something that is more numerical and is more rigorous in terms of Boolean logic and digital logic there. And we have, for instance, gone through some regulatory networks that we have studied in detail in the past, and we have been able to reconvert them into a little a logic structure in which each regulatory action is converted into some type of a Boolean operation. And well, this is again something very interesting, because now we can extract, or to get these gates, extract them from the natural context and put them in a different context to see whether we can manage bacteria to communicate to each other, because many of these gates involve not only the regulatory actions, but also metabolic transformations. So on top of that, well, and again, this is something that I will not tell you in more detail, but you can really go to the transcriptional regulators and try to reshape their logic by introducing their structure, protease cleaving sites, and at the end you can get a whole repertoire of gates that will maintain its natural logic, or they will display an engineered logic, depending on where you have put the protease cleaving sites. I'm sure I have to rush through this. I will be happy to tell you more about it if you are interested. But then at the end, what we would like to do is to see whether we can formalize this type of communication, in which the metabolism of one bacterium releases into the medium a metabolite that can be understood by a receiver cell as an input. And then we can formalize the release of this intermediate, and then in the shape of logic gates, and maybe at the end we can construct small circuits in which the whole consortium runs some type of logic operation. So as a very, very simple example, what I made with one of my collaborators, it was to put two Pseudomonas strains together, one of them converting toluene all the way into benzoate, and another consuming benzoate and sensing in the medium the presence of benzoate through a transcriptional regulator here. So what we were able to show by using, again, luminescent reporters and other types of tricks that molecular biologists do, is that indeed when you put bacteria that have been engineered to either release into the medium benzoate or to sense the presence of benzoate into the medium, then you get a signal where the two are together, and one can sense as a signal, as an input, the output of the other bacteria here. So this is still very, very incipient, but people that play with the idea of constructing communication languages between bacteria, for the most part, they bet on this phenomenon called quorum sensing. We are convinced that in nature, quorum sensing is important, but communication through metabolic signals is still more important. And we propose that perhaps we can capitalize on this metabolic communication to engineer some type of language to make consortia to develop some type of common language there. Okay, I think that that's basically what I wanted to tell you. Just to recap, so we're interested in communities. There are various issues in community construction. One of them is to make sure that the surface of bacteria can accept these adhesins. There's this issue of adhesins themselves. I think that the way to go is to play with this camel antibody story that I told you. And finally, when you have this geometrical structure, you have to put something inside, and one of the many things that you can put inside is perhaps to have this communication language based metabolic signals. And that's where we are at the moment, and we'll see how all these take us to. Thank you. Yes.
Can you tell me some more about the camel antibody and why it has evolved a single chain antibody rather than a two-chain?
Yes. Okay. So, well, this is obviously not my work, but the work of others. These camels were discovered by a Belgian group, by someone called Hammers, in Africa, and they have the theory that perhaps these antibodies evolved to counteract the effect of snake venoms. Okay, and you know, if you look at the structure of the antibodies, then you see that they tend to get this antigen binding or antigen recognizing loop of the structure. They try to get inside the active centers of enzymes. Okay, and this is something that we have exploited for other purposes. So apparently, there's a type of antibodies that might be connected to inhibiting directly the enzymes of others. Okay, so these type of antibodies are present in all the camelidae, and I'm not a zoologist, and also in squales, in sharks, and things like that. Okay, obviously we prefer to use camels than sharks, but these proteins are very, very interesting because they can be handled in the laboratory very, very easily. They have a very sturdy structures. they are very stable, extremely stable, and they are produced very, very well in bacteria. So that's something that simplifies our life tremendously. Yeah, but they are very, very good too. The problem is that you need camels, but that's another story. Okay.
So, Victor, is there any reason or is there any evidence why a structures community where you would have cell A next to cell B next to cell C, so sort of absolutely mixed, would be better than having a microcolony of A next to a microcolony of B and a microcolony of C?
Well, I think that is a question of a mass transfer and channeling of substrates and products between one step and the other. So intuitively, I think that having cells just hanging around, or even having colonies that are separated in space, might be less efficient. But I don't have any data, so that's a question that could be very interesting to see. And in fact, one of the questions, well, we have in the agenda 2 possible communities to check this. One is a community for degradation of orthochlorotoluene, and the other is a community for degradation of PCBs. Okay, in one case, and in both cases, we have 3 bacteria there. So one of the things that we're trying to check is exactly this type of situation. What is better, to have the 3 strains hanging around, to have them in close proximity but without any adhesion, or to have them really sticking to each other? And I have no idea what will happen. I know what I would like to have, but I'm not sure of whether we'll have that or not.
But I guess that's a typical modeling issue.
Sure.
That modelers should be able to model this.
Yeah, yes.
Conversion rates in
Yeah.
bacteria.
Absolutely. Yeah. Yes.
The Boolean circuits, can you comment on how robust is that description of what happens in the bacteria?
Okay. We're running into some terminology problems. So what do you mean by robustness of the Boolean circuit? I don't really get it.
So if I look at the Boolean circuit and I know what input is, I can compute exactly what the output will be.
Yes, well, yeah. Well, I'm sure I'm not a mathematician, so I'm not sure that I use the terms rigorously.
Okay.
But well, in biological regulation nodes and devices, you like it or not, the regulation is not 0 to all. I mean, you have all these intermediate stages and so, and obviously the capacity of modeling on the basis of zeros and ones is a kind of low resolution approach. Okay, so obviously you want to model exactly in more detail. You have to go into differential equations and so. But I have to say that compared to just a very soft description of the regulation versus having something where that you have zeros and ones, inaccurate as this might reflect reality, I think it's a big advance, right? So. And also depending on the promoter and depending on the specific system, then you have more or less stringent regulation and so. So that's another difficulty. One of the ambitions of synthetic biology, as you may know, is trying to start with existing regulatory nodes and try to make them as digital as possible. And this is something that in some cases is successful, in other cases is not successful. But this is the state of affairs at the moment. So if you say, are digital circuits robust in biology? My answer is today, no. Maybe in the future they will be. Right? Yes.
Do you know of examples of intra-species metabolically based communications that are studied? Could you say, you said a lot of people just say it's quorum sensing, but metabolism is important, and you're coming from the synthetic angle. Do you know? Are there some wild examples? I can't think of anything, like that.
Well, I cannot tell you like that, but that doesn't mean that they don't exist. Okay, so maybe I should really take a look at that.
There's the syntrophic consortia.
Yeah, but that's a different story in my opinion. I mean, in the syntrophic consortia, it's a kind of sequential degradation of something between A, B, and C. But I think that is that a communication or is just a necessity, right? So
it's a communication at some point.
All right. So so the limits between having just a syntrophic community or having something where metabolites are used as a communication signal, I'm not really sure, or whether it has been formalized as such. I'm not really sure about that. You have any example?
Well, no. Well, I was kind of thinking with the vitamin B symbiosis. It's not, at least with a system that Alison Smith's lab worked with and I work with as well. it's not clear. You know, we don't know yet if quorum sensing is involved or if the symbiosis can be completely predicated on just metabolic, you know, exchange. So I would say maybe the criteria would be if it's passive or not. You know, if the metabolite can stimulate like upregulation or.
Okay, so your question raises another issue that I find is very interesting. It's connected to the talk that Jeff Gore gave another day, and is you know, this signaling between bacteria and the common good story. Okay, so there are metabolites that never make it out. Other metabolites go out. That means that they can be captured by the other guys in town. So and I wonder whether there's a logic on that, or it's just a random occurrence. So some bacteria release some amino acids for no reason. They could use that amino acid for themselves, but they put that amino acid in circulation. So maybe the others can use that as a signal. I don't know, this is something that has not been really studied very well. So there's a metabolome that is always non-diffusible. There's a metabolome that can be diffused with no problem. And there's a metabolome that seems to work in proximity. So these are things that I think deserve some kind of attention. Yeah.
I think it's absolutely fantastic that they can do all these genetic manipulations, that they can shave cells and stick them together and so on. But it's a little bit hard for me to imagine how you would use that for practical purposes. So can you explain what actually you want to do in the end? So what I can imagine naively would be to have a sort of a chemical reactor or a chemostat in which this culture would be continuously growing, and then water polluted with toluene, for example, would be coming on one side and clear water would be coming on the other side. But these bacteria, are they supposed to be static there and not dividing, or because otherwise you would have to then separate them from what is coming out?
Yeah, well, you are running then into what is called biochemical engineering, right? So it's not only about developing the catalyst, but also to develop a process in which you make sure that the mass balance, mass balance and everything is in shape. So obviously the development of these say materials is part of the problem or the process, but obviously it's not the whole process. Okay, so in that respect you are correct. You also raised on as a side question something I find very interesting, and it's this problem of catalysis versus growth. Okay, because in the best of the worlds, what one engineer would like to have is a catalyst that is incredibly catalytic but don't grow, because if they grow then things start to get crazy. So that's an issue that perhaps can be tackled again through genetic engineering or through some type of genetic kind of trick through which to uncouple catalysis from metabolism and so. And this is something that is on top of the table of many synthetic biology groups. Not much has been done so far. Okay?
But is it possible at all to have a culture of bacteria that do not divide at all, but they just metabolize?
It depends on the reaction. Okay, so if the reaction is a passive reaction, you can even have dead bacteria to do the job. Okay, and there are, for instance, dealogenases that are produced by bacteria, and there's a bioremediation is a protocol to remediate some soils contaminated with some chlorinated compounds, and just to take dead biomass, mix it with bacteria and mix it with soil, and that's it. In other cases, where you use some type of redox input and some cofactor recycling and so, then bacteria have to be metabolically active, you know, and get this a window in which they are active, but they are not dead is sometimes difficult. Okay, but I think this can be tackled, and is being tackled again through some synthetic biology, where you can get deep into the metabolism and rewire this and that to make sure that bacteria are happy and producing their entire metabolism without growing. This is, in my opinion, a very, very interesting challenge. Okay, ladies and gentlemen, thank you very much.